So I think for this audience, I think a lot of you probably share the same view that we think that decoding the genomic sequence is almost a ideal problem for machine learning, right? So we obviously know the entire human genome, um, about 3 billion base pairs, which is a reasonably large data set. And we also have, at the same time, lots of genome-wide measurements for different genomic properties, right? But what is not directly observed are how are those two data are connected, right? How are those genomic properties are determined or dependent on the sequence. That's not something that we can directly observe from data. It is also, to some extent, hard to obtain from purely experimental approach. So that's an ideal problem to address through machine learning approaches to build sequence models that can model those different levels of regulations from the sequence, right? And I think another reason that's very important that makes sequence-based model powerful is that the sequence-based models often can capture the correct causality directions from the cause to the consequence, right? So this is not in general guaranteed for machine learning models. Like if you predict gene expression from histone mark, it's not clear which one is causing the other, right? But sequence, most of the time, it's on the upstream of those downstream regulatory elements. So it has an advantage in being able to capture the causal relationships so we can make use of the sequence models to make the prediction of effects of sequence perturbations and predict its expected consequence. And that's also often verified by experiment. So I think that makes sequence model a very powerful approach for us to decode the genome-based regulations, right? And today I will be focusing on our recent work modeling the multiscale 3D genome architecture, which is also our first model that can do the sequence modeling at the entire chromosome scale. And if we have time, I will also talk about another of our recent work that tries to develop a sequence-based vocabulary of genome regulation called sequence classes in the end. So to start on the 3D genome structure modeling, so we now know that our genomic DNA is highly spatially organized at different spatial scales, right? And a lot of what we learn about the 3D genome is coming from assays like HiC, where we can detect the 3D genome through sequencing approach in this way. Like we first can fragment the genome into different pieces, and then we ligate those fragments that are close to each other together. And then through sequencing, we can count how many of those pairs of fragments are physically close in this 3D genome. And we can obtain those matrices like this. So in this matrix, so both x-axis and the y-axis would represent genomic locations, and the diagonals would be representing the self-interactions with the same location or very adjacent locations. And the off-diagonal positions would represent the interactions between two genomic locations. Right? And we can plot those matrices at different spatial scales. And we see many different characteristics at different spatial scale. So at the most local scale, we can so see those individual chromatin loops interactions, including enhancer promoter interactions and also the CTCF-mediated interactions, right? If we zoom out a little and we see those famous topologically associating domains where sequences within the same domain tend to preferentially interact with each other, but not with the other domains, right? So we zoom out again, then we see again a different structure once we look at spatial scales beyond 1 megabase pair. So here you can see those checkerboard or plate-like structures, and which we can interpret it as that the genomic DNA or chromatin can be divided into mainly two compartments, the compartment A and compartment B. So the sequences in the compartment A tend to interact with other sequences in the compartment A, and sequences that are in the compartment B tend to interact with other sequences in the compartment B, right? And this compartment A and compartment B are also labeled by color here, right? So like everything in biology, that the structure is always highly relevant to function. And we know that the chromatin compartment is highly related to gene expression. Those compartment A are enriched in active genes, and compartment B is enriched in inactive genes or gene desert, right? And we know that the 3D genome in general is involved in regulation of gene expression, the DNA replication, and DNA repair, right? So we do understand some aspects of how the 3D genome depend on the genomic sequence, but it's also very incomplete. So we know about its dependency on the CTCF, and CTCF motif is important for TAD, topological association domains. But we also know very little about how the sequence drive, for example, the enhancer promoter interactions or Polycomb-mediated interactions, or those chromatin compartments. So I think it is an important question to be able to model all those different scales of the 3D genome interactions from the sequence. So that's what we want to do. We want to be able to start from the genomic sequence, being able to model all those different scales of 3D genome structure just by a sequence model. And so prior to this work, there have been awesome works that has already been able to model the 3D genome structure at the sub-megabase pair scale, including DeepC and Akita. So what we want to do is be able to further extend that ability to model interactions that are beyond 1 megabase pair scale. So finally being able to model all the different scales that we can model, that we can measure through high-C type techniques, right? And the first challenge that we have to address is, of course, the scale of the data, right? So if you want to model the entire chromosome and its interactions, it is a huge sequence. It can be more than 100 megabase pairs of sequence, right? And at the same time, the 3D interactions be represented by a large matrix at the highest resolution that we can obtain is also a very huge matrix that's almost infeasible to directly model, right? So to address this, the first idea is that we can take a multi-scale, multi-resolution approach to high-C data. So it's also a natural fit to high-C type of data, because in high-C, the longer the genomic distance, the fewer counts that you will get, and you can only measure the long distance interactions in lower resolution, right? So it's also how generally high-C data is represented. For the larger distance and long-range interactions, we visualize it with lower resolution, and with shorter range interactions, we visualize it with higher resolution. So we can model the genome using this multi-resolution matrices. So for the longer distance interactions, we represent it with lower resolution, and when we zoom in iteratively into the region of interest and use higher and higher resolution to represent those different spatial scales. So in this way, we can use a fixed size matrix to represent those 3D interactions at different spatial scale. And to be able to generate those series of multi-scale predictions, we also just generate a series of sequence representations at different spatial resolution, right? So the prediction goes in this order. We start from the raw sequence, and through a convolutional network tower, we iteratively generate sequence representations at different resolution. At the lower level, one sequence take input from only a local chunk of sequence, where at the highest level, one value can be computed from 1 megabase pair of sequence so that it is having much lower resolution, but summarize information from a much larger chunk of sequence. And when we make the prediction, we also use the sequence representation at the corresponding level to make the 3D genome interaction prediction at the same level. And also we iteratively make predictions for the smaller and smaller region that zoom in into a sub-region of the higher level. And we also use the previous levels predictions as input together with the sequence representation. So this allows us to actually make use of the entire sequence, even when we are making prediction for very local interactions, right? So this is one of the key ideas, but this alone still does not allow us to train the model. So the model is in this way is still too big to be able to process even a single input. So we have to have some like engineering tricks to deal with the size of the model, and it's mainly the size of internal representations, right? So there are some standard approach to reduce the amount of memory used in deep learning models, like checkpointing. So we need to modify that to make this work. So the standard deep learning model execution is those layer-wise sequential executions, and all the internal representations are saved for use in the backward path, right? So, but this requires a lot of memory because you need to memorize all the internal representations. And the checkpointing approach reduces the amount of memory that is needed by only save the internal representations at the checkpoint. So at the non-checkpoint positions, the internal representations are thrown away, but you can recompute it during the backward pass. So this trades some memory usage by about 2x the computational cost. But this is still not feasible for this kind of model, because even the first layer, we cannot compute it. We cannot compute even a single layer of representations in this model. So the checkpointing is not enough. So what we do instead is this horizontal checkpointing. So the idea is that we execute only a subset of the model in the horizontal direction. So you can only compute it for a subset of the sequence at a time. In this way, we still preserve the ability to make the same computation as before, but the amount of memory that we need is very small because of this hierarchical structure, so that the output of this box is very small, right? So in this horizontal checkpointing, it allows to basically scan the sequence and make our predictions. So we can still do the training as usual, but it reduces the amount of memory required to what can be feasible through a normal GPU, right? So with those tricks that we can now train the model, and this is an example of predictions on a holdout chromosome. So this is a prediction for 32 megabase per region, and each plot on the left is zoomed into the center region of the red plot, right, all the way to the 1 megabase per region with 4 KB resolution. So we compare with the experimental measurement, that it shows a pretty good consistency between the two, right? And we can also capture the different types of structures at different scales. At the local scale, we capture those topologically associated domains. And when we look at more than 1 megabase per scale, we can see those place-like patterns which represent computing compartments. So both are predicted by the model, and we do a more quantitative comparison across all of the holdout chromosomes. We do see a pretty good correlation across different spatial scales. And the last slide show you the prediction up to the 32 megabase per scale, and we can actually make prediction all the way up to 256 megabase per scale, which can contain the largest human chromosome, like the chromosome 1. And it also can make predictions of the interchromosomal interactions in these 3 scales, so that we can actually make predictions of almost all spatial scales that can be measured in this manner. And those positions that are grayed out are the regions that are unmappable and therefore missing in the experimental data. So one thing that's interesting that we find is that the model can make predictions for different types of 3 genome interactions as well. So we know that most of the 3 genome interactions that we observed in high-C are driven by CTCF-mediated interactions. Or you see that the model can also make predictions for other types of interactions, which are often more cell type specific, example, in this example, that these are predictions for some Polycomb-mediated interactions, which are marked by H3K7 methylation. And these are also interactions that are only observed in embryonic stem cell, but not in the skin fibroblast. And this is also correctly predicted by the model. And this is another example of, again, different types of interactions, so interactions between promoters and enhancers, right? In this case, it's also a highly cell type specific interactions, and the model also is capable of capturing much of the structures of the enhancer promoter interactions in a cell type specific way. So we now see that the model can predict all those different types of interactions and across different scales, right? And what would we use this model for? So I think the use of sequence models is most of the time it's not really to reproduce the data that we already have, right? So I think the power really lies in the ability to make predictions in making prediction for new sequences. So we can give new sequences to the model, and the model will make the prediction for the 3D genome structure for that sequence. So there are two areas of applications that I will talk about. So one is the predictions for the genomic variant, especially those large structural variants, which tend to have the largest impact on the 3D genome. And the other is to use the model as a way to generate hypotheses about the sequence basis of the 3D genome organization. So we know that a lot of diseases are caused by those large structural variants, and the effects are at least partially mediated through the effects of those structural variants in the 3D genome, right? So we collected all of those structural variants that has been previously studied with high-C or full-C data available. So we try to collect as many as possible and tested its in silico predictions result and compare with the observed result. So we have the full result in the paper. I just show one example. But actually in all of those cases that we see a very good consistency between the prediction and the experiment. So I think it is really already being very usable to actually make predictions for the structural variant effect when we don't have experimental data. So this is an example of a locus that several different structural variants can cause different phenotypes, right? So those deletions, inversions, duplications, and can cause several different phenotypes. And here we reproduce the 3D genome by just computationally delete this sequence and then make predictions for its 3D genome structure, right? So this is a deletion of this region, and predicted here is the inversion of this region that's predicted here, and it's a duplication. These two are duplications of this region. So in all of those different cases, there is one thing shared in common about the 3D genome change. So they moved this positive enhancer into strong interactions with a different gene. Right here, this enhancer gained the strong interaction with PAX3. This enhancer gained interaction with Wnt6, and these two gained interaction with Wnt6, and here it gained interaction with the second copy of Indian hedgehog. So in all of those cases, a new gene come into interaction with this enhancer, right? And those different genes that are interacting with the enhancer actually correspond to the different phenotype that's caused. So that's fully consistent with the experimental study. And also if we compare with the full-C-seq experimental data directly, you also see that the predictions casted in a full-C-seq format is also very consistent with the experimental measurement, right? So we think that the 3D genome sequence model-based structural variant prediction, I think is at a level that can be very useful for understanding those structural variants that we don't have experimental data before. And other than the applications to understand those structural variants, I think one very interesting question to me is that why can we make such prediction, right? So we don't, as I said, I think we don't really understand a lot of the reasons why how 3D genomes are dependent on the sequence. And we think that definitely the sequence model has captured a lot of those sequence dependencies, but how do we extract such information? So I think one way I tend to think about this is that we can think of us as kind of like geneticists that are doing virtual experiments on those virtual cells, right? So we can think of the sequence model as kind of like a virtual cell where we can mutate the genomic sequence and then give it the sequence, and it will make predictions for the consequences, right? So we can design genetic screens on mutating different sequences according to our hypothesis and see whether the prediction will have the expected effect, or whether what the prediction will tell us. We can refine our screen and screen design and make a new round of genetic screen. So this allows us to, but based upon, like, build upon those many ideas that geneticists has come up with to study the sequence dependencies of sequence models. So of course, I think for different questions, that different type of strategies has to be designed. So there's no one solution that fit all. So I'll describe two different strategies that was used to probe the sequence spaces at different spatial scales. So at a more local scale, we use this 10 base pair resolution mutagenesis approach to try to find all of the sequence dependencies at motif size. So the idea is very simple here, that we just want to mutate every single 10 base pairs and see which ones will affect the 3D genome, right? And we just did this in a way that is more computationally efficient. So instead of doing one 10 BP mutation at a time, we introduce 20 10 base pair mutations. And because of that those mutations that has the effect is sparse, we can actually, with specific experimental design, we can actually deconvolve those effects in the end, and we get back the effect of each individual 10 BP. So the effect of every 10 BP in this region can be visualized in this track, where when there is higher peak, it means that this base pair is 10 base pair is likely having a strong impact with the to affect the 3D genome. And the first observation is, of course, it is as expected, the strongest signals are coming from the CTCF, and all of those sites that with the strongest level of signal has a strong CTCF motif, either at that site or in close proximity, right? But what is interesting is that there are still sequence dependencies in the sites that has only a moderate level of 3D genome impact. So we look at those that are having moderate level of impact and exclude the CTCF binding sites. And now we see some cell type specific enrichment of motifs. And in the H1ESC model, we see that those non-CTCF 3D genome impacting sites are enriched in those PO5F1 and PO5F1 SOX2 fusion motif, and which is both of those TFs are reprogramming factors that we know are important for the biology of the embryonic stem cell, right? And for the skin fibroblasts and the AP1 family, the Forstrom motifs are highly enriched, which are also consistent with the known biology of the fibroblast cells. So we see that those transferrin factors might have a very cell type specific effect on the 3D genome, and those are only observed in individual cell types. So those are can create new hypotheses about how the 3D genome may be dependent on cell type specific transferrin factors at a local scale. And so if we move beyond the local scale and ask the questions about what are the sequence determinants of the chromatin compartment, which are something that we know very little about. We do know that this chromatin compartment is dependent on the gene expression, but at the sequence level, what really drives it is something that's hard to study. For some of the reason could be that it is hard to manipulate a small chunk of sequence to change the chromatin compartment. So when we are testing it computationally, we also see no single 10 base pair mutation can significantly change the chromatin compartment. Right? So we take a different screen strategy here. So instead of just introducing a small mutation, we insert a large chunk of sequence into a target location and see whether that can change the chromatin compartment of that target location. And the change can be just measured through the 3 genome prediction changes. And in this way, we can screen a set of target locations and source sequences from the tiles entire region. So we did it chromosome-wide, but here I just show you example in this 32 meg base pair region, where we have 9 target locations that include both some A compartment locations and B compartment locations. And the source sequences comes from the entire 32 meg base pair region. So each source sequence is 12.8 kilo base pair, and it tiles the entire region. So the effect of every source sequence in each target can be visualized through a profile like this. So different x-axis are different source sequence locations. So this can be considered as the P can be considered as source sequences that has had an activity to change the 3D genome. And we can see that the target locations are actually grouped into mainly 2 groups, which are corresponding to A compartment locations and B compartment locations, right? So at the B compartment locations, they are mostly sensitive to sequence with an A compartment activity that can transition the compartment from B to A, right? So those source sequences detected here are those with strong A compartment activities, and they are pretty sparse, right? And on the other hand, if we look at these other A compartment locations, which are sensitive to A to B compartment transitions, we detect sequences with strong B compartment activities, which are sort of complementary to the A compartment activity, but it's much denser and not as sparse as the A compartment activity, right? So to look at it further, we did several more refined screens. So one of them is that we look at what sequence scale is needed for the A compartment and B compartment activity. So we tune the insertion size. In the previous plot, it shows you the insertion of 12.8 kilobase pair, which actually let us down after doing this screen so that we know what is the right insertion scale. First, we actually did this screen to see what are the insertion scale that can cause a significant change. And we find that the compartment A activity requires actually very short amount of sequence that are around 800 base pair is almost sufficient. Even 400 base pair has some partial activity, right? And in contrast, the B compartment activity, it requires a pretty long sequence, at least 10 kilo base pair to have this activity, right? And what are those positions that have the A compartment activity? They actually correspond with the positions with transcription initiation activity, right? So they are essentially places where transcription start. And if we look very closely, we actually find that it almost center exactly, those positions with the A compartment activity almost center exactly at the transcription start site. So the model actually learned to capture those transcription start site this model does not use any data other than 3 genome. So actually learn to capture the transcription initiation site without using any data other than the 3 genome data. And these are just an overall summary that shows you that the sequence with compartment activity is more than 2 orders of magnitude stronger in TSS activity than the sequence without it. And also, we look at whether the direction of the transcription matters. And it turns out to be, at least according to the model, it does not. So for all of the target sites, it does not matter whether the transcription start site is transcribed in this direction or the other direction. in the same target location, they have the same amount of activity in changing the 3D genome. So another question that we're asking is that what are the size of the sequence patterns that are required for those compartment activity? So it can be that the activity requires 10 kp of sequence, but the underlying sequence pattern is just mononucleotide, such as GC content, for example. Then it does not depend on complex sequence patterns. So to test the sequence patterns that are needed for this activity, we use a permutation before we do the insertion. So before we insert the sequence, we permute it by in this manner. So we can segment the sequence into different segments of the same size, and then we permute the order of the sequences. So by those permutation, we destroy all the sequence patterns that are longer than the segment size, right? So if the sequence activity is preserved, we can say that the sequence activity is not dependent on the sequence patterns that are longer than the scale. So what we see is that for the compartment A activity, the sequence activity is destroyed after we permute to the chunks that are smaller than 100 base pairs, which indicate that the sequence patterns that are more than 100 base pairs are needed for the compartment A activity, which is sort of consistent with our understanding of how the promoter sequence works, right? And for the compartment B activity, it is very surprising that almost none of those permutations does anything to the compartment B activity. You look closely, even increase a little when you permute it to the 2 base pair level, right? So that seems to suggest that compartment B activity does not depend on complex sequence patterns, or at least it's not necessary to have the complex sequence patterns. And it's also consistent with the AT content. So the more AT that you have in this region, the higher the compartment B activity that you will have. This is also something that's previously observed. So, but we now have the hypothesis that compartment B activity might be just the default fate of the genome. So if there is no compartment A activity, maybe the default fate is just to go to B compartment activity. That's why the permutation may slightly increase the compartment A activity, right? So this is the wildest experiment that we did. I'm not 100% sure that it's going to reproduce in experiment, but I think that it is interesting to observe. So we just take a long chunk of regions, and then we permute them by two base pair chunks, and we see what happens after we permute the sequence. And we find that in the A compartment regions, if we permute the sequence, actually it changed from A to B compartment in those regions. It works in different A compartment locations. And if we permute in the B compartment, it does almost nothing. The sequence still stays to be B compartment, which is consistent with the hypothesis of the B compartment being a default fate of the genome. So taken together, I think this is the model that we hypothesize about the commuting compartments sequence basis. So short sequence at the TSS can drive widespread commuting compartment A formation, which is likely due to the large scale chromatin change that's induced by the transcription. And for the compartment B formation, it does not depend on complex sequence patterns beyond AT content. It doesn't mean that complex sequence pattern cannot induce compartment B, but mostly just that that all seems to be necessary for the compartment B formation. And it requires relatively long sequence without the compartment A activity to form compartment B. And coincidentally, about the same time, there was, so Job Decker's group developed an assay called liquid IC, where they chop the genome into different fragments and see whether the chromatin compartment structure is preserved. And what they observe is that if we cut the genome into segments that are shorter than 10 kilobase pair, the chromatin compartment starts to disappear. And at a level that's larger than 10 base pair, the compartment structure remains intact. So, which together with this seems to indicate that about 10 kilobase pair might be some important biophysical spatial scale that's needed for some mechanism of the compartment formation. So that's all of the story for the 3D genome. And I think the sequence model for the 3D genome is a tool that can be used for many different questions related to the 3D genome, and including the ones that I've talked about, from the predicting the structural variants to finding the effects of finding the potential hypothesis about the 3D genome organization. And we have the web server and the code and the models all available. And web server is mainly designed so that you can easily make a prediction for any of the structural variants that and yeah, without any coding that's needed. So do I have time for this second short story? About, I think maybe 10 minutes would be enough. Yes, okay. So I think I'll talk about another story. So here we are trying to just build a I call it regulatory vocabulary that can summarize the regulatory function of the sequence in a way that is interpretable, right? So now we can make a lot of predictions of different chromatin profiles, transcription factors, and histone marks. It becomes very difficult to actually have a global view of those. What are the integrity effects of those different chromatin profiles? So we want to be able to summarize in a way that is more interpretable and hopefully more relevant to directly relevant to its biological function. So this is work with amazing student Cassie Chen. So we can basically now use the approach to assign every piece of genomic sequence to a sequence class. So it can be visualized, the entire genome can be visualized in a plot like this, where each dot indicate a chunk of 4 KB genomic sequence. And the colors are based on the sequence classes, which group them based on their regulatory activity. So before I go deeper into this plot, I'll first talk about how we get here, right? So we first want to just build to get this picture of that we try to capture all of the regulatory activities. We try to build a model that can capture as much of the regulatory dependencies as possible, right? So we want to build the model that can predict most of the publicly available data. So in the same model, we used all of the profiles from the system, which process the publicly available chromatin profiles, and also integrated with the encode and roadmap, which taken together is more than 20,000 of different cis-regulatory profiles, right? And now we can make prediction for all of those different profiles, but we really need a way to be able to have a easy to interpret way to understand those 20,000 profiles. We want to summarize them into a smaller set of vocabulary that we call sequence classes. And to do that, we first just extract the sequences from the entire genome, right? So it's 30 million sequences that tiles the entire genome, and then we made the prediction for all of them. So in the end, we get the 30 million by 21,000 matrix that are predictions for each individual sequence. And now what we do to summarize this big matrix into a smaller number of dimensions that's interpretable, right? So I considered that we considered both the clustering and the matrix decomposition and those approaches. And in the end, we decided to do something that's simple, right? We just will use a graph clustering algorithm to cluster them into different groups. And we call those different clusters sequence classes. And we can also use that to give a quantitative score for every sequence. So for each cluster, we can determine its center and have a vector that represents the direction of that cluster. And then for every sequence prediction, we can project that onto the unit vector that represents each sequence class and get a projection score of the sequence prediction on each sequence class, so that we can quantitatively measure how each sequence is represented in the direction of each sequence class, right? So So with those, go back to the clustering steps that after we did the clustering and we visualize the 30 million by 20,000 matrix in a UMAP. This is a UMAP Disney combined strategy to get a map like this. And we can color those dots by the sequence classes. And some interesting organizational structure can be observed here too. So at the center, we see mostly those low signal sequences. So even though it is a small region, it contains a large part of the genome, which are just having low activity. And reading out at the peripheral, you have more sequences with more functional annotations and functional activity expected. So a large part of those map are different enhancer activities. So there are different cell type specific enhancers for the blood cells here, and there are also other tissue specific enhancer activity here. And there are some enhancers that are observed in multiple tissues that we call it the multi-tissue enhancer. And there are also those regions that are mostly called Polycomb repressed, and the transcribed regions tend to form its own sequence classes, and the heterochromatin has its own sequence class. And among the heterochromatin, the centromeres has formed its own class as well. And the two most distinct sequence class are the promoter and the CTCF cohesin binding sequences. So they are so distinct that in the visualization, it tend to be detached from the rest of the map. So with those sequence classes, I just want to jump to what we can do with it, right? So one thing that we can do is that we can, if once we have a mutation, we can now just quantify how much that the mutation will change the sequence class score, right? For example, a mutation may be able to change one sequence from a stem cell enhancer to a low signal sequence class, right? Which represents that it can strongly decrease the stem cell enhancer activity. Right. For every mutation, we can compute these scores in all 40 sequence classes. And here we did it for all of the HGMD non-coding pathogenic mutations. So basically all the disease-causing non-coding mutations that are included in HGMD. And all the mutations are represented here. And those that have a strong effect will be shown in the larger dots here. And those with a positive effect, which means that increase the sequence class activity, will be shown inside of the circle, and those with the negative effect, which decrease the sequence class activity, is outside of the circle. So we see that most of the sequence mutations are having, so the largest chunk of those are having an effect on the promoter sequence class, right? Which isn't surprising. Partly could be because of there is some bias of the study that focusing more on promoters, right? And there are also some mutations that are actually increasing promoter activity, which are also consistent with the prior knowledge about those mutations. There is a pretty famous mutations in TERT, which increase the expression of TERT in melanoma. And therefore, I think cause a consequence in the telomere and support the cancer growth. So in the non-promoter sequence classes, and we see majority of those are those that affect different enhancers. And what's interesting here is that we find the different enhancer sequence classes affected correspond very well with the disease phenotype. So even though that the prediction itself doesn't use any gene annotation. So it's purely just based on the regulatory effect predicted from the sequence. For example, for the liver intestine enhancer sequence class, we see that those mutations are predicted to have a negative effect, include those that cause protein C deficiency, factor 7 deficiency, and those are proteins that are produced in the liver, right? And for the erythroblast, like enhancers, those mutations that have an effect in aerosol blood, like enhancers, are causing phenotypes in the red blood cells, which are also consistent with the prediction. And also for the stem cell enhancer, those mutations that cause a decrease in stem cell enhancer activity are those that cause a developmental phenotype, right? So in almost all of those cases where when the sequence mutations are predicted to have a strong effect on the sequence class, it is consistent with the tissue type that it's predicted to impact, right? I think this is a good level of support for us to think that it is powerful in being able to understand individual mutations. And yeah, here there's no selection of mutations, but only selection based on the strong predictive effect that are shown. All the mutations with strong predictive effect are shown here. So I think last thing that's maybe worth mentioning is that those with the strongest positive effect are those that affect the CTCF cohesin binding sequence class. So some of them are known to actually increase the CTCF binding, but a lot of them are unknown, that we don't know what are the cause of those mutations. But it seems like they may cause a strong increase in CTCF cohesin binding, which may in consequence cause a 3D genome restructuring and therefore mediated the phenotype. So other than the interpreting individual mutations, that actually sequence class also happen to be a good choice for analyzing the heritability genome-wide on GWAS signals. So sequence class give you a set of 40 different partitions of the genome, which can just simply partition the genome-wide heritability into these 40 sequence classes. And we see that when we analyze all those UK Bellplan GWAS, we see that the blood enhancer sequence classes are enriching heritability from the blood-related traits and the immune-related traits, right? But only the immune-related enhancers are enriched in immune-related traits, and those liver and erythroblasts related to enhancer sequence classes are only enriched in red blood cell-related traits. And the brain and stem cell enhancers are strongly enriched in heritabilities for mental and cognition-related traits. And those multi-tissue enhancers are enriched in a group of pretty diverse set of traits related to body, lung, and blood pressure. And the promoter sequence class is one that stands out that is almost enriched in all of those different traits. So these are also, I think, another interesting observation that we can actually do this without using any gene annotation, right? So it's entirely based on the regulatory properties that are predicted from the sequence, and without using any gene information, you can capture the tissue-specific regulatory signal. So yeah, for the C and sequence class, we also provide all the web server and the models available. And I'd like to thank my team, and Cassie is the one who leads the C and sequence class study. Orca was done just by me when I was the only person in the lab in early pandemic, but now I have a team, and you'll probably hear more about their work later on. So yeah, thank you for listening. I'm happy to take any question if we have time.